Our first talk will be given by two people, actually. The Dr. Ada Cavalcanti from the Max Planck Institute for Medical Research in Heidelberg, and also by the Dr. Victoria Levario Diaz, who is currently a postdoctoral researcher in Dr. Cavalcanti's lab.
Thank you very much, Mathieu, for the nice invitation. So as you mentioned, I mean, we will split the time in two, because first part actually is about a technique that now we routinely use in the lab, and we established recently. But the protocol has been published, so you may find even a video about all preparation. And in the second part, Victoria will tell more about some recent development and some unpublished work with a more fancy combination and use of the Primo. So I will tell you a bit how we pattern hydrogels and use them for traction force microscopy in the lab. And the first thing is that it says a kind of motivation. Surface patterning serves many purposes. And in the lab, we do a lot of work with nanopatterning, actually. That's historically how we started. And then we extended to micropatterning, and now we combine both of them, both on soft and rigid substrate. The use of soft substrate is in combination with patterning is mainly finds purposes, of course, to measure cell-generated forces. And there are many ways to actually measure cellular forces, which are nicely illustrated here in this review with a 2D traction microscopy. This is what I will talk about today, where one can track the displacement of a substrate or fiducial markers, and then get information about this kind of force ranges. The 3D traction force microscopy is a little bit more complicated. We hope to get there in the near future to also track precisely cell-generated forces in 3D. And of course, something that is, I would say, in between, with a pillar displacement, with also microfabrication of this post and recording displacement also from cells. The traction force microscopy at the end, as I mentioned, is based on fluorescence microscopy, use of fluorescent beads that get really drugged by cells. And these beads are embedded in soft substrates like polyacrylamide. And one can really record how these markers get moved by the cell as the cell is generating forces. This is like a typical example of a fibroblast and what one can get by calculating at the end what is known from the elastic substrate, the fluorescent labeling of the beads, and solving, of course, is like with the linear elasticity problem, and get this kind of traction maps for a single cell, for example, here. The Primo setup that we use, and mainly Joel initiated this whole thing with the traction force on a Primo. Joel is sitting here in the Primo room, but unfortunately cannot be here today for the seminar. But our setup is actually unique in the sense that we also now combine it with a nano indenter system. So if some of you are also interested in how this thing can work, I'm happy to share our experience. And at the same time as we prepare this kind of materials, we can measure their elasticity also on the same microscope, and even probe cell mechanics. I mentioned to you this protocol has been recently published in this journal, so the video is also open access, and you can see all the fabrication steps. And what I wanted to share with you today is a little bit just the key concept in the protocol, so that maybe it sticks in your head roughly with the different steps. And for details, you can refer to the paper. So we use polyacrylamide because it's chemically inert, and so there is no unspecific interaction. But of course, PAA is not allowing covalent attachment of proteins to let the cells adhere. And so different groups can be coupled to PAA, such as like with the sulfosampa or NHS acrylate, and then in the end, one can really covalently mobilize proteins. The preparation, the classical preparation, is like with a droplet of this polymer solution and let it polymerize, and there is a thick layer of beads in the end. And if one is doing a confocal scan, you see all the beads basically across the entire material. And what JOVE actually developed is like a dual layer polyacrylamide substrate with the advantage like of having the beads trapped only on top of the gel. So where, in fact, I mean, the material mechanically is exactly the same, but only at the surface we have the beads, and then therefore it's easier also to reconstruct the displacement of the beads. And on top, actually, the reactive surface is a little bit different than what is classically prepared with the groups that I mentioned to you. And we mainly use the reactive aldehyde groups. And this is how now the confocal scan looks like with all the beads actually just in the upper section of the material. And this is how a cell looks on top of this carpet of beads. Another advantage is that besides making it easier for tracking is that you can use now the traction force microscopy on wide field microscopes and even with a relatively low magnification. And this is how the cells look like on these materials. This is an example here with different type of labels. And in the end, again, one measures the displacement triggered by this attachment of a cell on a carpet of fibronectin and the beads underneath. Now comes the patterning step. So having prepared, and this is really just a schematic with the key steps, but having prepared the dual layer polyacrylamide, actually, as the top layer is polymerizing, one can couple it to adhesive pattern glass on top of it. And by the fact that we have these reactive aldehyde groups, actually the fibronectin that is on the pattern gets completely transferred to the surface of the hydrogels. And this is why how one of our hydrogels look like with multiple cells or a single cell in a circle. And Joel also used the Primo to actually detach the cells, because one of the key steps in traction force microscopy, I was saying that it took advantage of the Primo, actually in the patterning with the light to disrupt adhesion of cells to the substrate and therefore release the cellular traction forces. And in this schematic here is like the kind of setup that he used. And we go from a deformed state or spread state to undeformed state where cells round up. And there is actually an induction of oxidative stress and release of tractions and later on cell death. The only thing to keep in mind is that it's a bit longer time than with the trypsin, so it takes 15 minutes to have this round state. But the advantage is that you can target different parts of the substrate and don't lose the cells all at once. So you can record the traction force in a sequential order in different parts of your samples. And this is our characterization. So as I mentioned to you, we have different ways for this piece of work. Actually, we use AFM, but one can use also nano-indenter. And we really took care of checking if all the different treatments that we did to our materials would alter the mechanical properties. And it's not really a huge difference, but one should really mention that the materials get a little bit stiffer as we deposit the matrix and we even do this UV treatment for the detachment. And these are two examples just to close up. One for a single cell, this traction force microscopy. So with a detachment here, it's like a nice fibroblast sitting here, and then after 15 minutes with a UVA exposure, it gets rounded up, and one can resolve and extract the traction forces here. Or one can really target a cluster of cells, for example, with a different size of 100 nanometer cluster or 300 nanometer clusters. So I've been saying this really in a nutshell. What I showed you is the preparation of this dual layer polyacrylamide hydrogels, and that these hydrogels are suitable for transferring of matrix proteins for micropatterning and to perform then traction force microscopy and use, in this case, the Primo also to have a more targeted recording of cellular forces. So I'm done with my part, and now I will hand over the word to Victoria. Actually, we are swapping the offices for Corona reason, so she will continue on something else. Good.
Hello. Yeah, my name is Victoria. I'm a current postdoc here with Ada, and I'm going to present some of the work that I've been doing, that I've been using, adapting the Primo setup that we have to do 1D micro-nanopattern integrin ligand surfaces. So as an overview of what I've done is this creation or fabrication of nanopattern surfaces that combining two techniques, which is the BCMNL technique, which is gold nanoparticles, where you can attach an RGD sequence to recruit alpha 5 beta 3 integrin of the cells and coordinate the movement front and back of the cells. So in order to do that, we're trying to tackle 3 different scales, the molecular scale, to see what is happening with the proteins that are involved at the nanoscale, because we're using, we're coordinating this movement at the nanoscale form, and also we are doing microscale because we are patterning some micron size with lines. So what we're trying to answer is, according to the integrin ligand spacing and density, if it can affect the coordination from the back and the rear of the cell, if we can modulate that. So as a general overview, this is the protocol that we've developed so far, where in the first day we have the nanopattern part with the gold nanoparticles done, and we also coat the surfaces with negative photoresist, so we can take it to the Primo to do a maskless UV lithography. We designed these lines beforehand using graphic software. We can load it to the Primo, and then we can to the computer, and then we can pattern it. So in this case, it's 10 microns width line. They're separated by 200 microns. And after that, we develop the lines, and then after we remove the gold nanoparticles that are in between the lines, the ones that are not protected by the photoresist, then we clean them, and then we do the RGD incubation to adhere the cells. We stain the nuclei, in this case with cDNA, and then we track the cells. We do overnight for 15 hours time-lapse imaging. These are the results that we have. Joel, as Ada previously mentioned, he set up the Primo system, so he has been helping me out to do the patterning. And we pattern lines where we have BCMNL spacing of gold nanoparticles of 50 nanometer lateral spacing between the gold nanodots. Then we have 80 nanometers also spaced gold nanodots, and 100 nanometers. So we want to see if by changing the lateral size of the gold nanoparticles, how it affects the migration behavior of the cells. And as you can see here with SEM images helped by Ioannis, we can see that we have the gold nanoparticles inside the lines, the pattern lines, and we remove all the gold nanoparticles from outside the lines. We also did some critical point drying to see the cells adhere to these lines with the gold nanoparticles, and we can see these protrusions, the philopodia who have integrins and are interacting with the gold nanoparticles that are biofunctionalized with the CRGD sequence. This is an example of how we did it. We did a time-lapse imaging in polarized light to see the edges of the cell, and we also, as I mentioned before, we labeled the nuclei with the cDNA, and we imaged it with the Sci-Fi channel. These are some videos that we have that we can actually, by tracking the nuclei, the position of the nuclei, we can also know about the retraction and spreading events of the cell. This is a representative cells, but I'm going to show you in this next sample how the Andreas Fink, which is a student here, developed a Python script so we can actually know the position of the cell nuclei, track the position of the nuclei, and we can extract the trajectory projected in a 1D line, and then we can have the displacement of the cells, and we can also have the directionality. So either it goes to the right or it goes to the right, to the left. For this, we analyze 50 cells per gold nanospacing, lateral spacing. Student Rebecca also helped us to analyze all these data that we had. And as you can see, when the cells are in a 50 nanometer lateral spacing, adhere to the biofunctionalized gold nanoparticles, the cells actually spend more time in the spreading phase. And with the 80, it's in the middle in between 100 and 50. As you can see, with the 100, we see more cycles of spreading and retraction. And they also migrate with a higher velocity as the 50 and the 80. We're studying these still events. We're looking that the displacement is significant. Is really significant, as well as the velocity and the total displacement of the cells. So after that, we also visualize the focal adhesions, and we try to quantify the size of the vincling clusters for controls. That the controls were some glass cover slip, also micropattern with the Primo system, but we didn't functionalize the gold nanoparticles with the CRGD motive. And as you can see, we have more vincling clusters with the CRGD, with the gold nanoparticles that have CRGD. We analyze also here for this case 15 cells per spacing. In summary, the Primo help us in making and fabricating our as well, Wenchan, who started this project, and the students who have helping me with Joel, and Alvéole, and of course, and the Max Planck Institute for Intelligent Systems.
Okay, thank you very much, both of you. Is there some questions? If you have some questions, you can ask them in the chat. I have some questions. So first, for the Dr. Cavalcanti on the first talk, I was wondering what's the main difficulty in recording focal adhesions dynamics in traction force microscopy?
Yes, one difficulty is, of course, I mean, if you really want to see the focal adhesion cells, there are lots of like details. And so one difficulty we had from experiences with the current microscopy setup to get good quality of fluorescent labeling of focal adhesions, really resolving forces in such a localized way. Still, we are not, it's not so optimal. One of the reasons is because of the optical properties also of the polyacrylamide. So right now, in fact, Joel is using elastomer that is higher refractive index. So it's really getting much better. So I would say one difficulty is really like the technical, yeah, with the material itself and the properties and getting good optics combined to everything.
Okay, thanks. We have a question also from Maria Hernandez Miranda. So it's, were there any problems regarding micropatterning on soft polyacrylamide hydrogels, as the nature of the material difficults this in comparison to stiff hydrogels?
Yeah, in fact, no, because I mean, the trick is really as top layer polymerizes to really also do the transfer at the same time. So it's a little bit like, that's the trick, yeah, to really trap the matrix protein as we're getting there. We're aware of all these irregularities, particularly polyacrylamide gels, the soft ones that are not really flat. But yeah, as I said, because of this kind of having the dual layer, that helps a lot, at least in our hands here.
Okay, so we have a question from John Yin to Victoria Levario Diaz, Dr. Levario Diaz. So it's your displacements tracked over 8 hours. What is the longest duration one may track such cell behaviors?
The longest duration of, I'm sorry, can you repeat the question?
Your displacements tracked over 8 hours. What is the longest duration one may track such cell behaviors?
The analysis, we did it for 8 hours only, but I've been working with several time lapses. So far we've reached 15 hours and they still, have several displacements. The total displacement, as I can put here, for each cell line, for each spacing, sorry, is around 50 microns for the 50 nanometers. That is all for 8 hours only. So we're now working with longer times to have more, to see if it changes.
Okay, thank you very much. So thank you both for your presentations. It was really interesting to see new methods to measure traction force on the one hand and to understand the cell migration mechanisms on the second hand.